Ladies and gentlemen, welcome to the special lectures of the day. Now let's welcome President Chi Hui Wong to deliver the opening remarks and give the brief introduction about today's speaker, Dr. Richard Lerner. Now let's welcome President Wong.
Okay, thank you very much for coming to the special lecture today. It's really my great honor and pleasure to introduce Professor Richard Lerner, who is currently Institute Professor of Scripps Research Institute. As you know, Richard was my boss and of course also my friend, because I'm getting old also, so I consider Richard is a friend now. Richard got his undergraduate training at Northwestern University and then MD degree from Stanford University. And after that, he spent almost his entire career at the Scripps, from postdoc fellow to faculty and member of the Department of Immunology and Molecular Biology, and then become chairman of the Department of Molecular Biology, and then director of the Institute, and then president of Scripps. So when I got there, that was 1989. He was director of the whole Institute, and then the Institute changed the name to the Scripps Research Institute, and he became the president of the Institute. So in his more than 25 years as a head of the Institute, he transformed the Scripps Institute into the largest biomedical science research institute, and also well recognized for the two graduate programs, one in chemistry and the other biology. And in his own research, I'm sure you are also aware of that Richard is a pioneer in the discovery, development, and implementation of combinatorial antibodies. So this discovery changed the way we think about the immunology system and the chemistry. And it's basically a kind of natural selection process in vitro. So his methods enable the creation of a large number of antibodies as a library to be screened, amplified, and optimized to target almost any molecules or any function, any reactions of interest. So with that accomplishment, he was recognized for many honors and awards, including, for example, the World Prize in Chemistry. Is that Park Davis Award, right? And then California Scientist of the Year and University of California Presidential Medal. And most recently, the Prince of Austria's Award. That's the highest honor in Spain. And he's a member of National Academy of Science and the Swedish Academy of Science. So I'm not going to go through all his remarkable career, and I'd like to ask Richard to deliver his talk. And the title is Chemistry for Large Numbers. And this title attracts a lot of people. I saw a physicist sitting here. Probably he made a mistake to come here. He thought maybe you were dealing with physics. So Richard.
Thank you very much. It's a pleasure to be here. I guess Taiwan owes a lot to us because we made a great contribution to the society because we had nothing to do with it. We couldn't stop it, but enabling and helping and encouraging and being the friend of Professor Wong, who's known worldwide, not only by us, but everybody for doing such a great job in Taiwan. And this you're lucky to work here. This is a truly remarkable place, and due to no small part to you, Chi-Wei, and then also great figures like you and Lee and other kind of people. You'll see in a moment why I chose that title, and there may even be something here for a physicist. But speaking of titles, I remember in the early days of the subject, I went to a symposium, and the speaker before me had toward a cure for AIDS, and the next speaker had toward a cure for obesity, and they had about 5,000 people in the audience, right? After the cure for AIDS, 2,500 left, and after the cure for obesity, another 2,500 left, and I had about five remaining. So the next year, I put a title, Anabolic toward a cure for AIDS and obesity, right? And then I had 5,000, right? we never done it yet. So I'm going to divide this lecture and sort of sit down and relax, because the subject is at some level complicated and at another level very simple. But I'm going to divide it into two parts. Part one, most of the components you will know like the back of your hand. But I'd like to weave them together in a way you may not have thought about them in order to set the stage for the new data. So if, as I go through it, you think to yourself, gee, this guy is just telling me everything. I already knew everything he said so far, right? Be patient, because that will only be the background to go into the more interesting things. That is, yeah. So we are going to talk. Can you hear me? The microphone. Oh, okay. Fine. We're going to talk about diversity, and whether you realize it or not, you have been interested and have witnessed diversity your entire life. And if you are interested in the physical sciences, for example, you probably have marveled at the fact that no two snowflakes are alike. And exactly how that happens is a matter of enormous interest, particularly to crystallographers and people who are interested in physical chemistry. To the biologists, diversity is equally important, and here's an example of that, because no two zebras are alike. So every zebra is different than every other zebra, and it's a whole field of developmental biology and pattern formation that worries about how that happens. We're going to talk today about diversity, but what we're interested in is mining diversity, particularly of the antibody molecule, for discovery and therapy, and we're going to do that by combinatorial antibody libraries. And I'll tell you in a moment what a combinatorial antibody library is, but basically it is a combination of the heavy and light chain that make up the antibody molecule, and using the combinatorial methodology, those libraries nowadays are very large, and they will have upwards of 10 to the 11th members. Now again, by way of background, this is how your immune system works. And basically, the fundamental principle is that you have in your body 10 to the 8th little packages. In immunology, they would be called clones. And each one of those packages makes one antibody. But the essential principle is that because that antibody molecule, or the gene for it, or the protein, however you like to look at it, is packaged, then the recognition on the surface of the cell and the replication are linked. So the immune system works basically because recognition and replication are linked in a package. The synthetic immune system, or the combinatorial library, has 10 to the 11th members, and instead of being 100 grams, a diversity system 1,000 times bigger will be contained in about 250 microliters. So that is the difference between the two immune systems, the natural one, the synthetic one, and you want to talk about it whatever we can. I'm not going to go into how it's made right now. And so essentially what we're going to talk about is the chemistry of very large numbers in a test tube. And as I'll say again in a moment, if you want to put your hat on for imagination, you should think about the fact, especially if you're a physicist and you don't want to think about antibodies, what could you do in your particular discipline if you had the control over 10 to the 11th different binding proteins? It's a very large number of binding proteins. Now, I dare say that everybody in this room has cloned something, or certainly knows what it is. And cloning is the art of taking something that's complex and reducing it to a singularity. So when you clone a gene, you're trying to get your hands on one gene out of, let's say, 50,000 genes. That's what cloning is, because in science what we like to do is we like to work with as pure material as possible. But look what happened here. What happened here is, because of the unusual nature of the immune system, we started with all these beautiful packages. Everything was packaged perfectly. Recognition, replication were linked. And then we took the whole thing apart. So no antibody anymore, or no antibody gene had an address. It's the opposite of cloning. And then we randomized everybody, put everybody in a bottle, and made 10 to the 11th combinations of heavy and light chain, almost none of which were the original pairs. So we ruined the clones. So the net effect is that one synthesizes an antibody gene repertoire that's 3 orders of magnitude larger than the natural repertoire, but the recognition and replication linkage are gone. It's important to think about. So the principles then that we have to think about is A, as I said to you, cloning takes complexity and reduces it to a singularity. Initially, and that's the operative word, combinatorial libraries convert singularities to complexity. So what must we do? We must return complexity back to a singularity and complete the whole circle. And that's done in a variety of ways these days. They're all simple systems. There's E. coli, yeast, spores, ribosomes, retroviruses, M13, and phage lambda. But you don't have to memorize all these systems. They're all wonderful little systems. But the key is that the library is repackaged So you reinitiated recognition and replication. So you're back where you started from, except in bigger numbers and in things that replicate much faster, such as phage. So this is what the system looks like, and maybe this will help take it away. So this is the M13 variety. This is a cartoon, and M13 is a micron-long rod with a single-stranded DNA on the inside, which has been tricked by us to carry the antibody gene. Okay. And then on the tip here is the gene 3 location protein, and there are 5 copies of that. And so the whole trick is that what you must do is you must get the gene inside and make the system express the antibody on the outside. So each one of the 10 to the 11th particles has a single gene on the inside, the antibody on the outside, and therefore in that micron-long package, helical package, recognition and replication are linked. So this is what it actually looks like in the electron micrograph. And what this is an antibody that was selected in the system to bind the hepatitis B particle. And there's nothing special about this antibody except it binds the hepatitis B, and since hepatitis B is a nice little particle, nice little round particle, it allows us to look at the proximity, the nature of the binding, and the stoichiometry of the situation. And these circles here, dense circles, they're the hepatitis B particle. And what you notice, take this away your head if you will, that every hepatitis B is attached to the tip of a phage particle. And the reason for that is because this is where the antibody lives. So inside this rod is the gene, and outside of the rod is an antibody. And usually you have one phage per virus particle here. Sometimes you have 2, but that's very rare. So on the average, one phage particle, one antigen binding event. So combinatorial antibody libraries then allow creation of a synthetic immune system in a test tube. You start with very large numbers. Since the process is carried out totally in vitro, it is scalable and repeatable. We use the principles of the natural repertoire, where genotype and phenotype are linked in a package capable of replication. So linkage and replication are important. You do not have to immunize animals anymore. We have been immunizing animals in immunology for 200 years. You do not do that anymore. A lot of people do, don't get me wrong, but you don't have to. And in some cases, it's the only way. For example, if you want an antibody to iron oxide, you can't inject iron oxide into an animal, but you can easily select antibodies that bind to iron oxide, okay? But the bottom line is we're fundamentally chemists, and we change antibody generation from an imperial exercise to an investigator-controlled chemical science. Now, today, you can sequence 10 million of these antibodies, and you can line them up in terms of chemical relatedness, all kinds of things, hydrophobicity, and understand what kind of chemistry in the antibody molecule allows for binding, and what kind of chemistry is forbidden or not preferred. This is important. There's a phenomenon in immunology called immune tolerance, which basically says that you cannot make antibodies to yourself. It's not perfect because there are diseases called autoimmunity. That's antibody to yourself. But by and large, you can't do that. Now, since all the antibody on the market today are antibodies to yourself, basically, because you're trying to perturb something in your body, it becomes very important to get around immune tolerance. So, the ultimate power is in the numbers, and I come back now before we go on to the more interesting things. This is the simple question I asked you in the beginning. If you had control in your own system of over 10, 11 binding proteins, what could you do? And one thing you could do is engage in the exercise of selection. And that's basically how everything I'm going to talk to you today works. So you start with big numbers down here, and by a selection process, which is like climbing a mountain, everybody drops off, or most everybody drops off, and then you have a couple winners, and hopefully these winners will be interesting. They will perturb cells, they will neutralize these viruses, they will block Ebola virus, they will block SARS, they will do all kinds of interesting things. Okay, so let me just illustrate the numbers to you, and then we go on to new things. So we wanted to find antibodies that broadly neutralize influenza virus. The operative word here is broadly neutralize, because as you know, influenza virus changes every year, and that's how it escapes. And so you want to find you want to engage in an exercise where you find an antibody that neutralizes many, many years back, and hopefully many years forward, because it is broadly neutralized. My purpose here is just to illustrate the numbers game to you before you go on. So there was an epidemic, as you know, of bird flu that occurred in Turkey. And the pediatrician who had seen these young children, most of whom were from farms and they were dealing with chickens and therefore got the bird flu, got very, very sick. This illustration of that, if there are any medical people in the audience. Here was the distribution in Turkey, and 6 of the children died. So this was not a minor infection. Then 2 other groups, the Crucell Corporation in the Netherlands deliberately made antibodies to bird flu. And in Boston, an antibody library, these are all libraries by the way, an antibody library was made from nurses who were taking care of patients with flu, not necessarily bird flu. Now, when these antibody libraries were analyzed, to the immunologists in the room, you see something very strange. Because if I were to immunize you with poliovirus the old way, everybody in the room would, as we know now historically, everybody in the room would make antibodies to poliovirus, but everybody would make different antibodies. So you will see your poliovirus, so to speak, in a private way. But the way you take care of poliovirus, and in terms of binding energy, and the way Professor Wong takes care of poliovirus will be different, but both efficient. However, in these cases, all the antibodies, in terms of germline, were basically the same, and all the mutations were the same. Even though the libraries were made from people from 4 different parts of the world. And as it turns out, as I'll now show you, the reason for that is that during the selection, remember I told you that's part of the process, during the selection, we put pressure on the system, and the pressure we put on is, don't show up unless you're broadly neutralizing. Okay, so that's all we wanted to see. And here are the crystal structures. These are not cartoons, crystal structures of the antibodies binding to the hemagglutinin. And you notice they're all the same. They're all the same. Historically, the influenza virus neutralization game is played up here. played out over these loops. Chile knows this system well, and it's very, very clever, the virus, because the virus attracts you up here to this loop or that loop or this loop. And it's more than happy for you to do that because it's a loop, so it can change. So, you chase the virus every year. Next year, the virus changes. You chase it again, and you keep chasing the virus. And probably for centuries, that game has been played. But here, there's not much variation allowed because this is the functional region of the virus. It's where fusion takes place. And when antibodies bind here, this cannot spring open, and the entry mechanism or the fusion mechanism of the virus is disabled. And that's why these guys are broadly neutralizing. Another antibody worked this way. And what it did was insert a long CDR3 into the sialic acid binding site, and therefore it's broadly neutralizing because that's the business of the virus, and you can't change that very much. You can change it a little bit, but you cannot lose the binding energy for sialic acid, otherwise you don't have a virus anymore. Now, the reason I showed you these examples, I could have showed you lots of other things, is because the frequency of these antibodies is about 1 in 10 to the 8th. So if you did 100 hybridomas, you would never see these antibodies. And in fact, that's the main reason they hadn't been seen before. They may have been seen once before by a Japanese worker, but it wasn't quite certain what he actually had. So I think the numbers are important. Okay, let's go to the new things now. That was the first generation of combinatorial antibody libraries. And in that generation, what you selected for was for binding energy or binding. And basically the idea was you had a protein, you had a virus, you had this, you had that, had a crystal, you had iron oxide, whatever, and you wanted to select antibodies that bound to it. So you would take a library of 10 to 11 antibodies, you would bind things 3 or 4 times. Today you would do it differently, you would use deep sequencing, but then it was different, you bind it 3 or 4 times. You get yourself between 100,000 antibodies, and now you sort through those antibodies to figure out which is the best. And usually best is defined as tightest binding. That's sort of what you want. You want the one that binds the best, right? Well, we will instead, as you'll see now, want antibodies that change the function of cells. We don't care about binding that much. We care about perturbing the function of a cell. So we'd like to change a red cell into a white cell, a tumor cell into a non-malignant cell, a fibroblast into an iPS cell, so on and so forth. The problem with phage, of course, is that it does not infect animal cells, and so you cannot do these things. So here's our old friend, the phage. And what you'd like to do is you'd build a system where the genes from here are portable, but in one step, into here. And that's the lentivirus. So if you take the genes out of phage, which you can do easily, and insert them into lentivirus, now you have an antibody library that will infect cells. So you've gone from a phage library to a library that's infectious for animal cells. And so you do this now. Forget this part. You take your lentivirus that contains antibody library, and practically speaking, you're now dealing with 100 million antibodies. you're not dealing with 10 to the 11th. That's just a practical issue. But now, these lentiviruses plus their antibody, they integrate into the nucleus. And now they start to make antibodies inside the cell. And your question is, what do those antibodies do? And do they do something interesting to the cell? One other small point, which turns out to be very useful, and that is that you usually get more than one antibody integrated into the genome, and that means you get bispecific antibodies whether you want them or not. Now, bispecific antibody for the non-aficionado is an antibody that has two heads, but they're different targets, and that's useful for certain things. Now you've got these cells. They have 10 to the 8th different antibodies in them, and the question is, can I take this nice whitey cell and turn it into all these different cells? So when I started, I asked you if you had 10 to 11 binding proteins, what could you do with them? And what we're going to see now is one thing you can do with them is take a cell like this and turn it into all these different cells. So here is a starter cell. This is a stem cell, so-called CD34 cells. And up until recently, what the world thought that cell does, and indeed what it does do, is it lives in your bone marrow and is responsible for the genesis of your red cells, your white cells, and your platelets, and maybe your macrophages, maybe not. So we take this cell and we get antibodies, I'll show you in a moment, that make this kind of dendritic cell or that kind of dendritic cell. Now this cell and this cell don't look like that cell, and indeed they're very different. So here's a case where you have an antibody that causes a cell efficiently to differentiate into something else. Now that something else may be on its normal lineage, so it's not unexpected, but the antibodies don't do that, except under this kind of selection. A different antibody makes white cells, different antibody makes platelets, different antibody makes M2 macrophages, different antibody makes red cells, different antibody makes tendons, and remarkably, as we'll see in a moment, an antibody converts these cells into neurons. So you have an antibody molecule that takes you from here to here, basically, right? Now this guy is not on the normal lineage of that cell. And there's no relationship with them. Now let me give you a feel for it. You know, they say a fingerspitzen. Let me show you what it looks like in the laboratory in a few examples. So if you take CD34 cells that are purified by cell sorting, and you put them in a dish, little plastic dish, basically nothing happens. They sit in that dish like little round BBs. You have BBs in Taiwan? What's the Taiwanese word, futon, for BB? Okay, little round nothings, okay, right? But if you put an antibody to the adiponectin receptor in that, about 4 days, here nothing happens. These cells begin to sit down, and they look like they have bubbles in them, big bubble wrap. But the reason you see these bubbles is because these cells are beginning to accumulate these lipid droplets in a peripheral fashion. And when you look at an electron micrograph of them, you see a couple things. You see, yes, the lipid droplets, but an enormous accumulation and synthesis of new mitochondria. And along with the synthesis of new mitochondria is made this uncoupling protein 1. And basically, this has converted that CD34 bone marrow cell into brown fat. And what the cell is doing now, instead of making red cells and white cells, what it's supposed to do, it's making heat. So you turn the cell into a heat generator, basically, right? Here you put this antibody in, you put this one in, and after about 4 days again, about 50% of the cells don't look like this anymore. They begin to sit down. They begin to take this morphology on their tips, and they begin to look like this. And these cells are staying with actin, and this happens to be the growth cone, neurotubulin, the nucleus, and many other classic markers. So you went from this cell to this cell, and it's a neuronal cell. So that's 2 cases. This antibody happens after the fact. We know what it is. It's an antibody to the integrin alpha 3 chain, and you don't see the bubbles, and you don't see the neurons, these long neurons, but instead you see this. Now, can you see? Tell me if you can. We'll turn the lights. Can you see that these cells are starting to make these little feet? Can you see those little black feet? Okay. And that's because these cells are going to crawl around the dish. And the reason they're going to crawl around the dish is actually they have become dendritic cells. They're quite proper antigen-presenting dendritic cells. Now, in every case so far, we've talked about converting one cell type to another one, and that was from a stem cell. This is a stem cell. Now, well, I didn't go into details, and I deliberately did not go into detail. We can certainly discuss it, and a lot of this is published. But in most cases, when you want to do a selection, you build a selection system by fooling around with the receptor and so on and so forth. And so the question we had most recently is, why bother with that? Why build a selection system? Why not just infect the stem cells straight away? Put them into a soft agar or something like that, and look for a phenotype. And by the time you get 20 phenotypes, you'll have everything. You know, how many cells are there? So here's the beginning of this. This is the same stem cell, but you infect here, and you get M2 macrophages. You infect here, and you get a cell I haven't thought about since medical school. It's a tenocyte, and it's the cell that makes type 1 collagen, and that's how your tendons are made. You infect here, and you have an antibody to the UNC5C receptor. Now, this is a famous receptor. This is a receptor. We didn't go into this experiment to get an antibody to that receptor. We just got it. And that receptor is the receptor that guides, tells axons, to be anthropomorphic for a second, how to go to the right connection, okay? And however, in our case, what it does is it causes this cell to become that cell. So now you're making neuronal cells, right? And this antibody, interesting antibody, it's to the discoid-like receptor 1. Its normal ligand is collagen 1, but this antibody causes this guy to become an osteoclast, a bone reabsorption cell. So it's quite easy to convert one cell into another cell. Now, given, I added this slide, but given the fact that we had some discussions before, this is one iteration of the system. And you take something, and what you're going to try and do here is you're trying to take one receptor and put 100 million antibodies in the cells, so that each cell has one receptor and a different antibody. And then you're going to hook the whole thing up to a beta-lactamase reporter gene. The important thing is it's an autocrine system. An autocrine means, as most of you know, that the ligand and the target are in the same cell. And then you go through a cell sorter, and everybody who turns from blue to green has an agonist in it. And this we don't have to dwell on. This is just a case to show you that the antibody and the receptor are in the same cell, and they co-localize in the membrane. Now what happens here? What happens here? So you start with a population that looks like this. Then, if you have 10 microgram per mil of antibody, it looks like this. You see all these neurons sitting down here, and they look like this. And they're really interesting cells, because what they do is, they're very long cells with a nucleus and a prominent growth cone, and they start to grow toward each other, and then they don't like it, because they're in the business of making proper connections, and this is wrong. So in accordance with proper neuronal behavior, they say, wait a second, not you, and they make a turn. The important point is this cell does not look like that cell. Furthermore, this is not on that lineage. That does not belong on the phenotypic lineage. And so what's happened here is that the antibody has taken a receptor that's used for something else, and caused the cells, bearing that receptor, to turn into brain cells. We look like this. Here's just a scanning EM of that slide, or a slide like it. And you see you still got a few round cells, but you've got all these cells that look like this. And that's a beautiful growth cone. Beautiful growth cone. Now there's a lot of genetic work to take you from the receptor we just saw, or the little round cell, to this kind of cell. And then when you stain it with all the markers, you get this remarkable cell with the actin distribution on the tip of the growth cone, neurospecific tubulin here, and the nucleus here. There are many other markers. I won't go through them all these markers, and all these markers, and it tells you that that cell is not a CD34 cell anymore. It's a brain cell. Okay. So what do we learn? It's one thing to make things, it's another thing to learn. And the antibodies taught us that one receptor can activate two different lineage outcomes, and the result depends on the mode of activation. Now, as chemists, what we believe is the cell can read dwell time. So it knows off rate, it knows recycling, so on and so forth. But it's not prepared for an antibody. It's prepared for a normal physiological receptor. The antibody hits it, probably binds too tightly, turns on the signal A too strongly, B out of proportion to other signals, and that signal says, don't become a white cell, become a brain cell. And that's what's happened here. Now here's the most important thing of all. What about cancer? Unfortunately, all cancer therapy today depends on killing cells, and the kind of killing you're interested in is selectivity. And so you're more or less successful in how effective you are in eliminating the shrapnel. So you want to only kill the cancer cell, you don't want to kill anybody around it, and you know, all the kinase inhibitors and all the chemotherapy and all, they're never perfect. That never happens, okay? But the Holy Grail is actually to differentiate cancer cells into less malignant cells or even end-stage phenotype. So if you could take a cancer cell and turn it into a toenail, that'd be a good thing. You know, you may have a toenail growing out of your forehead, but you would take that, you know, instead of cancer. Why should you think this is possible? Well, first of all, now maybe 40, 50 years ago, there were nuclear transplantation experiments. And those were done mainly by Beatrice Mintz. And what she did is she took a nucleus out of a cell that had been cancer for 200 passages, put it in an animal, killed the animal, put it into the egg of a new mouse, made a mouse, got a mouse that was born, and that mouse did not have total body cancer. The mouse had erased, I guess we would call it epigenetics today, but that mouse had erased all the cancer phenotype. So in her particular situation, cancer was an erasable phenotype as long as you knew how to do it. And her way was pretty gross. You know, she had to born it again. He had to reborn the nucleus. So that's one thing. So it's possible. Cancer cells are likely to have the stem cell phenotype. So why not perturb those stem cells? That's one of the real problems in cancer, to become something else. And finally, as I told you, we have shown that antibodies can induce differentiation and transdifferentiation of cancer cells. So why not use these antibodies to turn these stem cells into something else, and maybe the whole thing will become non-malignant. Now, we got from the bone marrow of a 60-year-old lady who was suffering from acute myelogenous leukemia, her cells. And these cells are very, very vicious. They have completely replaced her bone marrow. And when you put them in culture, they look like that. They're obviously stained here, a monotonous sea of unattached floating round cells. To the clinicians in here, if there are any, this is an M1 leukemia, it's very stem cell-like, and about 96% of the cells in her marrow are myeloblasts. However, when you mix these cells with one of the antibodies, they sit down like this. By the 5th day, about 50% of them are neural cells. They look like this or that. You see these are presynaptic vesicles. So these are proper neural cells, let's put it that way. And instead of looking like this, they look like that. And these cells have converted into these cells. And if you stain them, they have TUJ1, they have an active cap, they have GAD1 and MAP2. Those are markers of neurons, neural cells, let's put it that way. And I put this in here to show you that you see the leukemic cells, the remaining leukemic cells, they don't make these neural-specific markers. Only their DNA stains, as it should. So these cells are quite proper neural cells. And so what has happened here is an antibody has taken a vicious leukemia and turned at least half of its cells into neural cells. Now, in leukemia, you look at these bone marrows. I mean, I used to look at them as a student, and you get horrified because the whole marrow is replaced by these nasty-looking cells. But they're not the problem, actually, except for the crowding they do, because they're not renewable. They're going to die anyway. The problem is that 2% of them are stem cells. And they are going to drive, they're the well from which all this trouble happens. So if you don't care about all these guys. If you convert that 2% into these guys, you've gone a long way toward taking care of leukemia. Now, I'm not going to dwell on this except to tell you that we can do the exact same experiment for peptides. So probably, I don't know, in 50 years since Merrifield or whatever that number is, Chiwei, the scientific community has made, what do you want to say, 1 million peptides? Futan, how many peptides have been made in the history of peptide chemistry? 1 million? Let's say. We study 100 million at once. So here's a case we talk about later where we wanted to make a GLP-1 that was biased. That is to say, in GLP-1 activation, there's 2 pathways. One goes through cyclic AMP, and one goes through the beta-arrestin pathway. And we wanted to make, that's what the natural peptide does. We wanted to make a new peptide that goes only through this pathway and does not go through that pathway. And so here's our old friend, and I showed you before, and that was my reason to show you that in this situation you have 100 million antibodies and 1 receptor, and in this situation you have 1 receptor and 100 million peptides. And so you could select for a peptide that does something unusual. In the parlance, it would be called a biased peptide because it activates one pathway completely and not another pathway. And that leads to some really interesting physiology, which I won't go into. Okay. It's really ordinary kind of stuff, right? Let's take up the Ebola epidemic, or an approach to Ebola we were talking about in lunch. Don't worry, I'm coming to an end. Okay. So, I tried to say to you that the most powerful selection system you can have is an autocrine selection, because you got the receptor and the ligand in the same cell. That's not totally true. The most powerful selection you can have is prevention of cell death. There's no selection system that's more powerful than that because all the waste disappears, right? So here's an example of that. Selection of antibodies that protect cells from death. I'm going to tell you a rhinovirus story, but it might as well be an Ebola virus story. So you take, the trick here is, or the fact here is, that human rhinovirus will kill 100% of HeLa cells in 24 hours. So if you put human rhinovirus into a culture of HeLa cells, you come back 2 days later, everybody's dead, basically, right? However, in this experiment, you infect with an antibody library of 10 to the 8th members 2 days before you infect with rhinovirus. You come back 2 days later, and you have to work very, very hard to convince yourself there's a single living cell in that culture dish. But you have faith. You have faith in the antibody molecule. So you take all the antibody genes from whatever's still attached, and you do it again. You reinfect, okay? And now, you feel a little better about it, but you're not sure, okay? And you do it again, and you do it again, and you do it again, and by the time you're at the 5th passage, the rhinovirus can no longer kill the cells. Now, every cell in the culture is alive. And it looks like this. This is really a poor man's slide. So here's what the culture dish looks like, a flask, in the control, and you see a beautiful painting. Not such a beautiful painting, but Paula Foley likes it. But anyway, because you hold the flask up and you can see right through it, right? Because there are no cells on it anymore. However, if it's infected with the antibody molecule, you can't see through it. It's all blurry because the cells are not dead anymore. The antibody's protected it. Now, you sequence those antibodies, now the winners, and you know who they are. And I'll tell you now, it's no reason to be surprised. Out of the 10 to the 8th, you got 2 antibodies that won. Both those antibodies were against the cysteine protease that is encoded by the rhinovirus genome that is necessary for proper replication of the rhinovirus. One of the antibodies neutralizes the enzyme activity and the other does not, but it keeps the virus from the manufacturing well that it lives in. But now you know who they are. So you go back to the library. This is very instructive. You go back to the library and say, how did you show up? What were the kinetics of your appearance? So you start out here, and this is around, I guess, 0. And I can't see the number, but what is it? 0.001% or something like that. So the winning antibody, these are the 2 winners, are there in a very, very small population. This is by deep sequencing. And then as you go along, the diversity shrinks and the winners begin to show up. And this is based on 7 million reads. And by the time the entire culture is protected, 27 and 28% of the antibodies in there, the antibody genes in there, are encoding the protective antibodies. So that's what the kinetics of protection looks like. If you do it over again, but this time you use the membrane-bound library that I told you about, you don't get the cysteine protease. What you get this time is you get ICAM1, which is the rhinovirus receptor. So the parallels to Ebola are obvious, and you're going to select 2 kinds of antibodies. One just neutralizes the virus because it neutralizes the glycoprotein, and another that blocks the entry of the virus into the cell, and that's just a question of format. For entry, you use membrane-bound format, and for intracellular, you use the intracellular format. Okay, the final point. Like differentiation, development responds to events at the cell surface. I don't know if anybody here ever thought so, but if you consider the Yamanaka factors, differentiation and development don't work in this way. one day a cell wakes up and says, I think I'll turn on the 4 Yamanaka effectors. I mean, that's not what happens. Events of the cell surface begin a cascade, okay. Since antibodies reacting with the surface receptors can induce differentiation, why not development? What's the difference? What's so special about development? Development is sort of fancy differentiation. So, what about doing this kind of experiment? You take embryo fibroblasts and you start with Yamanaka factors, you put in all of them, and then one at a time you leave out a Yamanaka factor and you put in an antibody from the naive library. And when that antibody is successful, you leave out another factor, and another factor, and finally, there are no longer any Yamanaka factors, only antibodies. So here's just a case to show you how it was done that way. And if you leave out Nick and Sachs, right, you get these things that look like pluripotent colonies. You pick them and you dissociate them, you get a lot of colonies, okay? So, so you're making these colonies that stay with classical markers of IPS. They look like they're making all the germ layers. You see that there? Including beating heart, okay? Which looks like this if you blow it up. So now you've got IPS courtesy of the antibodies. And then you make this. Or this. And that's a chimeric mouse. That mouse has 3 Yamanaka factors and 1 antibody that is causing it to happen. We've now done it over again, and now we've replaced OCT4. So you have 2 Yamanaka factors and 2 antibodies, and we're on the way to have, we'll soon, I think, have 4 antibodies. So, combinatorial antibody libraries. They allow creation of a synthetic immune system in the test tube. Importantly, you circumvent self-immune tolerance. You depend on large numbers to allow selection of rare events, and you can generate research antibodies this way, or therapeutic antibodies. As I showed you in flu, you can identify targets. You can very quickly make agonist antibodies. You have new tools for the study of differentiation and development, and importantly, a gain of function method. It's not a loss of function. It can be, but for the most part, it's a gain of function, like gain life, gain neurons, gain what you like. And here's who did everything, right? So I began by showing you some structures, and all of those were done by the long-standing colleague of both Chiwei and me, and Wilson, the great crystallographer. The very first man to make these phenotypic selected libraries is Hongkai Zhang. But then the membrane bound was made by, where is he? GZ, we call him Sig. He calls himself Sig for cigar. I'll tell you a funny story about him. So I'm done anyway, almost done. Okay, so he was came for the first time from China to the United States, and it happened that we were having a it just happened. We were having a lab dinner downtown. Downtown San Diego is still a little rough. So he wanted to get an ice cream. It was the first event in the United States. So we were walking around the corner, And I said, by the way, what should I call you? He said, what do you mean, why should you call me? You know, this is my name. I have a name like everybody else. You call me by my name. I said, okay. We walked a little further, and there was a drunk who passed out of the gutter with a whiskey bottle. So I said, you know, he used to be a student of mine. And he said, well, what happened? I said, I can never remember his name. So he said, okay, fine, just call me Sig. Okay. And that's how he got that name, and now everybody knows him like that, okay. He's a wonderful, wonderful scientist. And many other people in the lab. All the work I showed you on the mice was done in collaboration with Kristen Baldwin, who's a card-carrying stem cell biologist, and her student, Joe Blanchard. And this has nothing to do with it at all, but it's a paper we just published in Science, and Chiwe knows Jiang Zhu, and maybe Raj Grover, who solved the structure of this really interesting antibody-binding protein. I thank you for your hospitality, I thank you for your attention, and give you all a scientific rose.